Good morning, everyone. It's a great pleasure to be here. Today we have the pleasure to receive Agustina Undabarrena from Chile. Agustina is a biologist, and she did all PhD and PhD, and up to now is also postdoc fellow with in Beatriz Câmara, which is a very good friend of mine, at the University of Universidade Técnica Frederico de Santa Maria in Valparaíso, in Chile. And she, just to give you an idea, this is one of these young, brilliant scientists, I would say. She got for her PhD in 2017 the best thesis in Chile, just to let you know. And she wants, the one thing that I really admire in her work is that she can transit from the bench to the in silico for bioinformatics. This is not very common for the people, and Agustina do this very well. And she had already several publications, and the main topic is always actinomycetes from marine environment, and she's doing this for a long time, as I understood, since the undergrad studies. And so, Agustina, thank you very much for your acceptance to be here with us. I will be here, I will close my camera, and I will be here in the background if you want something, and then the people can do the questions at the chat in YouTube. Even in Portuguese, we can, or in Spanish, we can translate it to everyone afterwards. Okay, good presentation, and thank you again.
Thank you, Alex. I will share my screen. You can see it?
Yes.
Yes, yes.
Okay, good. So, good morning, everyone. My name is Agustina Undabarrena. As Alex told you, I've been working with Bea a long time ago, and today I'm going to talk about insights into Rhodococcus and RPSs, a comparative genomic story that mainly was a side B project of my PhD thesis. So for those of you who don't know what we do, we actually study actinobacteria from the South Pacific, and we are in the search to find novel antibiotics because this resistance that most pathogens have emerged the last decade. And to do so, we study first the marine actinobacteria that we collect from marine sediments and also marine sponges along the Chilean coast. Then we sequence their genomes, and we do so to find the biosynthetic gene clusters, and I show you here how they are grouped. They have a lot of different genes, and you can see here in blue the genes for the biosynthesis. Yes, sorry.
Could you close this bar on the top and bottom? Yes, thank you.
Yes, okay, and maybe the laser is better there.
Yes, it's okay like this. Thank you.
Okay, thank you. So the biosynthetic gene clusters have different kind of genes, and in blue you have the genes for the biosynthesis. Here in light blue, the genes for the tailoring reactions, that are the genes that are involved in adding post-assembly modifications. Genes for the immunity, so this bioactive compound don't kill the host. And also genes for the regulation of the biosynthesis, so we have can have a negative or a positive or both. And genes also for the efflux or transport of the molecule. So I've been studying the biosynthetic gene clusters of the category of non-ribosomal peptide synthetases. And this particular synthase make peptidic bioactive compounds. They can be linear or they can be cyclic, and also they can have another modifications. So we are trying to understand the biosynthesis to look for these new bioactive compounds, and hopefully we can contribute to make another novel antibiotics. So we have three main areas understanding the actinobacteria from the South Pacific. First, we try to analyze the marine microbial diversity, and to do so we perform a lot of taxonomy and phylogenomics. Then we perform also genomics from the sequence of their genomes, and we perform similarity networks that you can see here, just to analyze the biosynthetic gene clusters. And also then we perform metabolomics. We grow the strains in different media. We perform LC high resolution MS. Also, we make similarity networks of the molecules, and then we try to identify the structure of the compound. So those are our three main areas of our research. And as I told you before, over the years, we have been sampling Chile. So this is our country, and all this is the ocean, the South Pacific Ocean. So it's a very vast coastline that we have to explore. And we did so, we sampled different very pristine areas, like for example, in the northern part of Chile, the Chañaral Island. And in the southern part, this is actually the northern Patagonia of Chile, we sample also the Cumau Fjord. Then we sample also islands, so Rapa Nui Island, maybe you know it, it's very well known for its moai. And also Valparaiso Bay, because it's where our university is, so we have a really close access to this part of the ocean. So in these places, we have taken marine sediments and also marine sponges, and this is just for you to look that we have retrieved a different biodiversity from each place. Actually, each place is looking very, very different in these pie charts. Some of the genera are found in all of them, but some are really exclusive from these locations. So that is telling you that it is still worth to explore and to isolate taxa from actinobacteria, and mostly if they are marine, because we know a little bit about marine actinobacteria and almost nothing about actinobacteria from the South Pacific. And this is just to have like an overview of all the biodiversity that we have collected through the years. We have 32 different genera comprising 20 families, and you can see here that the most abundant are Brevibacterium, Streptomyces, and Brachybacterium, but we also have this small pie chart that has all the rare actinomyces that are also very interesting to explore. If you want to see more about this, you can go to this publication where are all the details, not only the biodiversity details, but also the assessment of the bioactivity of these strains. Okay, so today I'm going to focus my talk in this pristine and unique place that I have a special interest for, because it was the place where my samples from my PhD came from. It's the Cumau Fjord in the northern part of the Chilean Patagonia. And especially, I'm going to talk to you about one strain that is a Rhodococcus strain HCA8F. So why I choose this strain? So this strain first was isolated from an island from marine sediments and 22 meters depth. And it showed a lot of bioactivity when I test these results in the lab. So here you can see that the Rhodococcus strain is at the middle of the plate. And then here you have another that are model strains, like for example, Salmonella, Pseudomonas, Staphylococcus aureus, Escherichia coli, and Listeria monocytogenes. And you can see how the growth of the Rhodococcus is able to inhibit the growth of these model strains. For example, Escherichia coli is almost fully inhibited. Also Listeria and Staphylococcus aureus like partially inhibit. So we want to explore this a little bit more. And then we perform culture extraction of the Rhodococcus strain using different organic solvents. And you can see here that we retrieve activity against these model strains in the ethyl acetate solvent, using the ethyl acetate solvent. So this was a very bioactive strain that prompted our attention, and we want to deeper understand how this strain was able to produce these bioactive compounds. So that's why we performed genome sequencing of this strain. We did this with the PacBio platform, and it was really good to use PacBio because we were able to close the genome. We have one chromosome that is fully complete. And also we were able to find another unitype that is a plasmid. And this is a very interesting unitype because it's a linear megaplasmid. And also most part of it, almost 800%, is hypothetical proteins. So this is a very novel plasmid, and we wanted to keep investigating the genome features of this strain. And also Rhodococcus is very well known for its catabolic metabolism. So once we have the genome, we wanted to explore if we could see by genome mining if this strain has the potentiality to also be involved in catabolism. So for example, we looked and we find a huge number of monoxygenases, for example, enzymes of cytochrome of P450 and also deoxygenases, also a lot of different routes. For example, this that I show you here is the biophenyl, and we were able to see also if this pathway was active because we grew the strain on a minimal plate with biphenylazole carbon source, and we were able to see that the strain actually grows. So this strain not only has the pathway, but also it has an active pathway because it was able to grow in the presence of biphenylazole carbon source. And this is a very well-studied phenomenon in our laboratory. And to cope with all this stress, this strain also has several genes for the oxidative stress response. And for example, the typical catalases, superoxido demutases, and alkyl hydroperoxido reductases, but also some new ones like bromoperoxidases and chloroperoxidases that are specifically found in the marine environment. Okay, so we have this very interesting strain I'll show you before that has a lot of bioactivity and also potential to produce, to degrade different compounds and to cope with the oxidative stress. But our remaining questions were, okay, this such unique strain and with such unique genome features can also have unique biosynthetic gene clusters. What can we know about its anabolism? And then also in the chemistry, can this biosynthetic gene cluster be translated into the identification of molecules harboring novel chemistry? That is our final aim. And the involution part, can we identify the main mechanism of the Vtc distribution across the genus? Because this was something that was not explored, and when we look how much was in literature about the Rhodococcus, we found that was very, very little regarding the biosynthetic gene cluster distribution. So with these 3 questions on mind, we started another new project, and we use the Bigscape and Corazon pipeline that you can go to the paper that was published last year. I have it here to, if you want to see more details, but mainly what you do with this is that you analyze a lot of genomes, then you use anti-smash to detect the biosynthetic gene clusters. Also, you can compare them with the MyBig platform, where you have the validated biosynthetic gene clusters, and then you use a tool called Bigscape that actually it groups biosynthetic gene clusters and make similarity networks. So you can find also in these networks some reference biosynthetic gene cluster that come from MyBig, and also the anti-smash predicted ones. And you can compare them with this biosynthetic gene cluster that is from the reference. And by understanding these network structures, you can actually see how your VGC connects to the others that can be known or unknown. And in a further step, you can also analyze the networks with this tool that is called Corazon. And what Corazon does actually is that looks within a network and see if you can find some correlation with the phylogeny. So you have here the biosynthetic gene clusters, and you can actually see the genes that are in the closely related species and which are absent and which have been duplicated or lost during the evolution. So we use these tools, but we apply them to the Rhodococcus genus. And that's why we first have to perform a pipeline, bioinformatic pipeline, because to use this platform, you start with genomes, right? And when you go to NCBI and you download all the genomes that are there about Rhodococcus, you can find about 300 genomes, more or less. But when you look at them, they are standard draft versions. So for us, it was really important to develop a pipeline, an in-house bioinformatic pipeline for the filtering and selection of genomes of good quality. For that, we performed several bioinformatic criteria and also applying biological criteria. We were able to filter these 300 to 110 genomes, and we have these genomes with good quality to perform comparative genomics, because then otherwise you will be having conclusions that are over-assumed because you have very draft genomes and you cannot compare them in terms of biosynthetic gene clusters. So to do this, we apply a manual check, and first we filter all the genomes that were that have more than 200 contigs, and then we use CheckM. That is a platform that checks for completeness and contamination of each genome. We use also OrthoFinder to analyze the tree, and if the group of genome branched together, then we apply another step with Annie to see if they were redundant genomes. And then also we select the genomes by the Bigscape distance, because we were focused on understanding the biosynthetic gene cluster. So then we also compare the genomes to see if they have redundant biosynthetic gene clusters. And by applying all these pipeline, we were able to finally retrieve 110 genomes. And with this information, then we perform a phylogenomic analysis. So the 110 filtered genomes have more than 1,000 biosynthetic gene clusters and 700 NRPSs. And with all these genomes, we have the phylogenomic analysis that I show you here. So we have mainly 4 major clades. Clade 1 here is in orange, clade 2 in green, clade 3 in purple, and clade 4 in blue. And our strain that I showed you before, Rhodococcus HCA8F, groups here in clade.
Agustina?
Acho que ela caiu.
Uhum.
Ela foi desconectada. Vamos esperar um pouquinho.
A Agustina, então, teve um...
Ela está entrando de novo.
Está entrando de novo. Teve um probleminha de conexão. A gente já está retomando aqui, ela já está entrando na sala novamente. Então, a gente já retoma. Enquanto isso, vocês podem ir organizando as perguntas de vocês para a gente poder, já na sequência da apresentação dela, fazer alguns comentários, né? Acho muito interessante essa parte, né? Até uma das coisas que eu fico curioso, por não ser muito da área, é quanto tempo será que levou para fazer essa descida de 300 genomas a chegar a 110, né? Uma das coisas que eu talvez venha perguntar, porque para mim é um trabalho manual bastante denso, me parece, né? Mas eu não tenho noção quanto tempo precisa para uma coisa dessa, né? Mas é um negócio muito interessante mesmo. Sorry for the Chileans online. I don't know if you understood what as I said. I was wondering how long it takes to have this from 300 genomes to 110 genomes. How long does it take to proceed this manual check? Okay, is coming.
Acho que ela ainda não entrou.
Não, ela
tá entrando nesse.
Fazer o microfone dela, eu acho.
Vai ter que repetir os últimos.
Os últimos. Beatriz just said that she understood my Portuguese. That's good, Beatriz. Beatriz, there's a funny story for yesterday. I didn't know that Brazil was playing against Chile in football. I don't know if you are aware. Probably most Brazilians don't, or much from here. And sometime in the night, Beatriz sent me a message, and no, it's not possible. And then I was kind of wondering, something happened with Agustina, that probably she came tomorrow, and so. And then I asked, what is it? And she said, no, that's Brazil made a goal against Chile. Okay, Bea, I didn't know that. At the end, it was 1-0 for Brazil, as I understood. Is that right, Bea? Bea is the supervisor of Agustina in Chile, which is my sister friend, let's say like this, a mix of sister and friend. We met each other in Germany when we are both in PhD course. I don't know if Agustina wrote here. Okay. Can you hear us, Agustina, or not? Just to maybe she has to check in again. Just a moment. Okay. 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 Because Beatriz is writing down there. Okay. Okay, she will try again to connect. Okay. Agustina, if you hear us, just yes, good. She went out now and she come back, probably to work now. So if you have some comments in the chat, just let us know. I was trying to talk about football, but Beatriz is not really in the mood, I understood. Ok. Deixa eu ver aqui. Só mais uns minutinhos de paciência, pessoal. Ela está tentando conectar de novo. Por algum motivo que ela achou que tivesse conectado, ela não viu que ela tinha caído. Foi uma coisa curiosa. Ela seguiu seguiu falando, em princípio, pelo que eu entendi dela aqui no chat. E aí ela...
É porque ela tava só com... Ela tá com a tela dela, só ela não via...
É, porque a gente não via a gente aqui, né? Daí quando eu tentei falar, ela... Ó, ela tá de novo agora.
Tá, autorizei.
Ok. Yeah, but I know you're not really in the mood. Okay, I understand that. I understand that. Can you hear us, Agustina?
Maybe she's coming yet.
Yeah.
Doesn't need to wait a few minutes, few seconds.
Ok, mais uns minuticos, gente. No, we can hear you. We cannot. Your mic is in mute, Agustina.
Can you hear me?
Yes.
Yes.
Okay, good.
I just let you have to share it again. And the slide, you are only to us the one that you made this manual check from 300 genomes to 110.
Oh, I was going forward and forward. Sorry.
We try to advise you, but I don't know. It is strange that you didn't see it. I tried to talk, but you are not able. You can share again your screen, Agustina. Sorry for that. But
sorry, everyone.
We don't know what happens actually.
Actually, Bea sent me a message, and that's why I noticed, because otherwise I was talking here.
Okay, good.
So, can you see the screen now?
Yes.
Yes, sorry about that.
This is the slide one. Okay, thank you. I will close here again, and good presentation.
Okay, so I will say this slide again because I don't know actually where I drop off. But the thing is that we download all the genomes from NCBI, and we apply a bioinformatic pipeline for quality control of this genome. And you can see here the pipeline. Actually, first we apply a manual check, and we discarded every genome that has more than 200 contigs. And then we use another tools like CheckM, OrthoFinder, and ANI with the aim of discard all the genomes that are redundant. And that's how we end up with 110 genomes that are of good quality to perform further comparative genomics analysis of Rhodococcus. Okay, so with the good quality genomes, first we perform taxonomy, and we did a phylogenomic tree with OrthoFinder. And you can find here that Rhodococcus mainly has 4 major clades. You can find here clade 1 in orange, clade 2 in green, 2A and 2B, clade 3 in purple, 3A and 3B, and clade 4 in blue, 4A and 4B. And our strain that I showed you before, this very interesting strain from the northern Patagonia, clades here in clay groups with another marine strain that is called Rhodococcus erythropolis PR4. They are the 2 marine from clade 4B. And in terms of ecological distribution, most Rhodococci come from different isolation source that you can see here in this square. This is the legend of the isolation source. And we found that there was not a clear correlation regarding the isolation source and the phylogenomic clades, with the exception of clade 2b that you can see here has a high number of strains that come from plant-derived ecology. So with this in mind, we tried to see first if we can find some relationships with the strains that were marine, but we found that there were a little of them. They are like 8 or something like that. So we don't have enough information to actually look for what is happening in the marine environment. And we encourage other authors to please sequence more marine strains so we can compare our strain and we can understand how these VTCs are distributed in the marine environment. But anyway, we move on and we use Bigscape and Anti-Smash to perform the Vtc Similarity Network that I showed you before in the pipeline. And here you see the network where each node is a different VTC, and they are all colored by the category, and they are connected also when they share a row distance below 0.6. That means that where they are similar, they are connected. And with that in mind, we can make another conclusions. For example, that the NRPS is the most abundant category within the network, as you can see here in blue. In bold black label, here you can find the biosynthetic gene clusters that come from our strain. And also we were able to notice that you have different patterns of evolution. You have, for example, this one that is a balloon-shaped structure, really, really close. And this is telling us that it's represented mostly by BGCs that are highly conserved across the genus. And this is not the case, for example, for the NRPSs that shows a more diverse pattern of evolution. And this is telling us that these biosynthetic genes shows a high genetic divergence with a lot of gene expansion and recruitment events that mainly comes from genes of primary metabolism. So we wanted to study the NRPSs a little bit more, and that's why if you take this network and clear all the nodes that are not NRPSs, so if you take only the blue ones, you can find this network that is an NRPS network. And here the color represent GCF. So gene cluster families, or GCF, is when you try to identify with Anti-Smash and also Bigscape if there is a node or a biosynthetic gene cluster that has a known compound that is involved in the production of a known compound. So then you can make assumption that all the nodes that are related to that one can be also producing a kind of similar molecule. But for us, we couldn't assign and identify GCF because the similarity scores were too low. And this suggests that mainly there is a vast group of NRPSs, but they are mostly uncharacterized. So we have here a lot of potentiality to find novel chemistry. And here we can also retrieve 44 GCF, and in the network, all the GCF that has more than 10 nodes are colored. So in terms of the network structure, here we can see that, for example, traits that are conferring a higher fitness represent a defined GCF, like for example this one, that is a network where all the nodes are present in GCF9. And in the other case, traits that are still being tested to fitness represent this kind of network where you can see a highly interconnection, for example, between GCF1 and GCF5. We tried also to color the same network, but now with the clades that I showed you before from the phylogenomic tree. So here you have the code color, and you can see, for example, the network that I showed you before, that was all the nodes were from GCF9. now we can see that they all came also from strains that are grouped in clade 4b. So here we could observe that the NRPS distribution was specific for certain clades, and there is a strong correlation with the phylogenomic clades rather than with isolation source. And this is suggesting that the NRPSs are being transferred vertically through the genus, which is highly controversial because you can see that for other genus, mostly the dynamic of biosynthetic gene cluster is the horizontal gene transfer. That is the most common. So this was our first novelty, to actually see that the biosynthetic gene clusters are being transferred vertically. Okay, so then we perform a presence-absence barcoding. So here you have all the clades and all the strains, and if this genome has a biosynthetic gene cluster that has representative within these gene cluster families, you can see, for example, dark color, and if it's not present, you can see the absence or a white color. And in this figure, we could observe that it was a streaking feature between GCF1 that you can see here in orange and GCF5 that you can see here in green. And they show a pattern that when one was present, the other was absent, and so on, especially in clade 4A and 4B. So we want to analyze this pattern better because we identified that these gene cluster families were interconnected. And also our strain that is here in yellow has a representative from GCF1. So we took this NRPS as a model to understand what was happening in between GCF1 and GCF5. And this cluster is the one that I show you here, is NRPS number 5 from GCF1. And you can see that is mainly composed by a left arm, a middle section with the biosynthetic genes, and also a right arm. This is a mega cluster, and I am going to explain to you what these parts are involving in producing differently. So first, in the left arm, we apply also corazon to use these genes as a query and to see whether they were also present in the other strains, but also to analyze their co-occurrence pattern. That means that if, for example, this gene is always together with this gene, then we put a geometric symbol to say that, okay, they are both present, so this is like a biosynthetic unit because they are both present, so that means that they are working together for something. And when we make BLAST, we analyze these genes, and we could see that, for example, these are transporters, are involved in the transportation of a molecule of 4 carbons. When we look a little bit more, we can see that, for example, here we find the regulatory genes. They were also together always, so that's why we put another geometric symbol here that is a cross, and we found that this is the DNA binding protein and this is the kinase response protein of the regulator. And then we have these 5 genes here, and they are all involved in the modification of this 4-carbon molecule. And then we have these 2 genes here that we group as an inverted triangle, and they are also involved in the transfer of this molecule to another step. Then we have the middle section that mainly encompasses 3 biosynthetic genes. We have here the predicted amino acid that each one is incorporated, and you can see that we have 17 amino acids in total, but they are mostly unknown because the prediction was too low to assign a respective amino acid. And also these 3 genes have unique domains, as I show you here, a C-started domain that has the ability to incorporate another thing that is not an amino acid, and this adenylation standalone domain that also has the ability to incorporate another monomer that is not peptidic. So these two domains are in charge of adding novel chemical diversity, and we think that this cluster is not mainly peptidic and has another features also. And on the other side, on the right arm, we grouped the genes similar to what I explained before, and we found that these genes here, the triangle ones, these genes here, are involved in the synthesis of a chloramphenicol-like molecule that is being modified with the addition of a nitro group. That is mainly because of this gene here, AURF, paraminobenzoato N oxygenase. And we also could see a peptidic antibiotic transporter here, these three genes, and a lipase and esterase. So with this in mind, we have the genome features of the cluster, and then we want to see how this cluster that I showed you before, that is complete in this upside of the figure, is being distributed across the phylogeny. So I told you before that our strain was located within the clade 4. And the way that we did this is that if these biosynthetic units that are formed with these genes were present, then the geometric symbol that I show you here has a color. And if some of these genes are missing, then you can see an empty geometric symbol. And with this in mind, we analyze all the structure of the VGCs that these strains show. For example, if you can see these parallel lines, it means that the genes were found, but they were in another place of the genome. So this was not a cluster, or they were not contiguous. And with this analysis, we find that NRPS5 that I show you here is a unique biosynthetic gene cluster, and there is no other cluster that looks like it, even in closely related species. So we wanted to analyze this not only in terms of genes, but also in terms of domains and modules. And we found here that the 3 NRPS genes that I told you before, here is our strain, and you can see a complete module within the same color. For example, this green and this green, they are different modules each. And you can see that the first NRPS is the most conserved one, and the diversity is mostly given by the second and the third NRPS gene. And also we saw another important feature, that is this adenylation domain that I told you before. That is a standalone adenylation domain in white, and we don't know actually if this was a step in evolution where this module was the domain of the adenylation domain of this module was duplicated, and then we can observe here 2 adenylations domain, or actually this was the first scenario and 1 adenylation domain was lost, and then we find here the complete module. What we do know is that these adenylation domains adds more chemical diversity because has the ability to add another monomer that is not a peptidic one. So that's another interesting feature of our cluster. And in this comparison with these strains, we could also observe that there is no other VGC that is identical to the one that we have, supporting further evidence that our VGC is unique within the clade. Okay, so then we have a very nice, unique biosynthetic cluster, but we wanted to explore if this strain, if we can link this biosynthetic gene cluster to a molecule. And that's why we perform the OSMAC approach, that is called one strain, many compounds, in which you grow the strain in different media. You perform fermentations and crude extract analysis to see if they have some activity. And we use the model bacteria that I showed you before. We use these 5 different media, and we could observe that was main bioactivity in ISP2 with artificial seawater, that is this media here, and this other, and this other. So we took these three that was the most bioactive ones, and we sent it to Fundación Medina in Granada, Spain, for the replication. And what they do there is actually they analyze the crude extract and they compare it to the in-house databases by LC high-resolution MS, and they detect if there is a known compound in such a mixture of the crude extract. And that's what we did in Fundación Medina. And you can see here the results from the LC high-resolution MS. We observed 3 different peaks. Here you can see the UV spectrum from this peak 1, peak 2, and peak 3, and also the masses. And when they match these compounds with the on-house database, they could identify peak 1 as chorinosine 1, peak 2 as chorinosine 2, and peak 3 as chorinosine 3. So you can find here the structure of these compounds. And this was a family of molecules that was really discovered in Corynebacterium, but was never found before in Rhodococcus. So this was the first time that we were detecting chorinosins in a strain from Rhodococcus. Okay, so as a graphical abstract, we found a very interesting strain from the northern Patagonia of Chile. We performed genome sequencing. We also did some comparative genomics and phylogeny with other strains that we download from the NCBI and we filter to have high quality data. We performed with Vtc Similarity Network to have insights into the dynamics of the biosynthetic genes, mainly in NRPSs. Also, we performed genome mining with anti-smash to have this unique mega cluster that I showed you before was very unique within the clade with the tool of Corazon. We perform also crude extracts and fermentations and the replication with Fundación Medina, and we found that this cluster was mainly involved in the production of chorinosins that are structurally related to the chloramphenicol compound, and that's why we observe such a nice bioactivity. So what I show you here today are in 3 different articles. You can go on them to see more details. This is the first one, the second where we describe the Rhodococcus genome features, and in the third one that is where all the similarity networks are, and also the Vtc and Corazon tools are applied. So you can go there to see more about this work in these articles. Taking home messages, so I hope that I could explain to you in the genomic part why a comparative genomic pipeline for analyzing VGCs was developed for us, how we group NRPSs into GCF, and this revealed mostly uncharacterized families, and how the NRPS pathways are correlated with phylogeny and not with isolation source, and that means that they are vertically distributed. In the chemistry part, how NRPS5 is a mega biosynthetic gene cluster composed by a left arm, a middle section, and a right arm, and the right arm is involved in the synthesis of a chloramphenicol-related molecule that is called chorinosins, and chorinosins present bioactivity in Rhodococcus crude extract. And in the evolution part, I hope I show you that NRPS5 is a unique biosynthetic gene cluster within its clade, that is clade 4, and that the left arm and the right arm are the most conserved along clade 4, but the variable region is given mainly by the NRPS genes, and this is resulting also in a variable peptidic core with further modifications that are novel chemistry to the biosynthetic gene cluster. So overall, the genomics, the chemistry, and the evolution of this biosynthetic gene cluster represents novelty, and that's why we were very interested in this cluster. So I just want to say thank you to my Actino team. In blue, you can see the people that work with me in this project: Bea, Andres, Leo, Richie, that is now in the UK, and also Paco from Mexico, and Lalo from Santiago, that help us with the bioinformatics pipeline. To all of them, thank you so much. And also thank you for listening to me, and I will be glad to answer your questions. Obrigado.
Okay, thank you, Agustina. Very nice talk. Very nice talk. There are some questions already. I would like to start with one that once you disconnected, I was wondering, and I also talked to the people that how long it takes to do this work manually. Let's say from 300 to 110 genomes, and someone said in the chat of YouTube that it took, depending on the computational work or performance, you took 1 week. I found this very fast. I think it is much more time. I'm wrong.
Well, for us, it was much more time. But I think that basically was because it was the first time that we did such a filtering. So this is very unusual actually, because sometimes you just download the genomes from NCBI and they have a good quality and you can further go through your comparative genome results. But for us, was not the case. We tried and we find, like, in the middle of the work that we need to apply a filtering pipeline, and then we go back and we start applying all these criteria, biological and also the bioinformatic ones, and we took months, I think, and also in between the correction and the manual check with the redundant strains. But I think that in the end, even if there was much time, it was really, really helpful, because at the end we could find results that we could actually link. You know, it's very difficult when you are trying to analyze a lot of data and then you focus on one and you try to link that one to another thing. So I think that for us it took a long time, but it was truly and definitely a good way to go, because then we can find something interesting at the end of our work.
Yeah, indeed. Thank you. We have some questions. We just put it here on the chat inside our room. Could you see this?
Yes, I see
yes.
that.
Yeah, the first question is from Janira. Que bom que tu tá aí, Janira. Janira is a known from she works also in this field, as I understood. She asks, as a perspective, do you intend to test the data generated associated with recombinant technologies, chemical synthesis, molecular engineering, and bacteriocin BGCs, and so on?
Oh, yes, we would like to do that very much. Thank you for your question. Actually, as I told you, this was like a side B project of my PhD. And then in my postdoc, I was learning how to perform genetic engineering, mostly in Streptomyces, but the technology can be also applied to Rhodococcus. And as a second step, we would like to perform maybe disruption of this cluster and to see if this molecule that we observe is also lost, and why not the overproduction for also the purification of the compound, because we believe that there is not only cryonesins involved, that also they are being taken together with the NRPS to form a bigger molecule, and we are not seeing the bigger molecule right now, so we want to explore this a little bit further. Yes.
Yeah, actually, I missed that. She continues with the questions afterwards, and also she asks us another strategy to test biological potency, stability, pharmacological properties in order to guarantee the potential activity of these BGCs.
Yes, of course, but with the purified compound. I think that is a step further in our research. We are now starting to improve our chemical pipeline also, and maybe yes, in the future we can test all the things that she says, and we can also see those properties in our compound.
And closing with Janira's question for the moment, and she asked also if you have found some bacteriocins BGCs
in this specific genome, or is she asked globally?
No, I think it's specifically. I would understand.
Okay, yes, we did. We find, I think it was 2 clusters of bacteriocins in this genome, and we also observed different kind of biosynthetic gene cluster of bacteriocins in our actinomycetes. Definitely, it is worth to check them because they are also can give bioactivity. But I focus on NRPSs mainly to do this work, so I didn't analyze the biosynthetic pattern of distribution of bacteriocins.
Okay, and then we have a question from Nicolau. Nicolau, he said that it's an amazing talk. I would like to hear about your experience with the big slice. I have tried to use big slice for comparing BGCs from fungi, and I found that while the tool is amazing for close related BGCs, the tool cannot properly link distantly related BGCs. For example, the one you only only the backbone and the additional gene are conserved.
Yes, well, for this research, we use big scape, not big slice, for this specific Rhodococcus. But we are now using big slice also. And the thing is that big slice is very good when you are comparing a high number of genomes, but a much even higher number of genomes. And big scape is more accurate. So I think that it is good to first use big slice to have like an overview of the diversity of the biosynthetic gene clusters that you can find in a genus or whatever you're looking for. But then it is also good to use big scape because the prediction is more accurate, and the relationships that you can have between when one node is connected to another one could be easier to interpret. So I think that, yes, that's the main problem of big slice, but also big slice is a very recent tool. So hopefully they will also work on improving that feature.
Okay.
For us, it worked well to complement big scape results with Corazon. So maybe my advice will be to, if he wants to try use Corazon, it works really well to understand these backbones that are conserved along a genus. So for us, it worked really well to use both.
Okay. So Nicolau, that's a tip for you. No. Thank you, Agustina. So Diego said that's nice and detailed bioinformatics works. Congratulations. He said one thing that fascinates me, actually, I'm not really in the field of bioinformatics, unfortunately, up to now. It's how do you see the interests or the importance is clear, but how do you see the interest from companies, pharma companies, for example, to do this, let's say, upside down search for the bioprospection, let's say like this? How do you see this, or how do you feel?
Oof, it's a very rough question. Actually, I think it's really hard that companies actually get involved. I think that there is a good science there to show them, but they are really, like, I think, afraid because over the years they have put a lot of money in this, and you have to try and error a lot just to come up with something that is good for pharmacy, you know? So there is a lot of time also involved. So for example, the genetic engineering that someone was telling before, you can be just years to be able to actually modify a biosynthetic gene cluster, and then there are also years to purify a molecule. So I think that the main drawback is that you need a lot of time, and we need those molecules now, because actually it's the problem that we have currently. So I think that it will be very nice that pharmaceutical companies actually get more involved with what we are doing in academia, because we truly think we can collaborate more and we can help them to guide the bioactivity bioprospection.
For sure, for sure, for sure. This is very interesting, because once you have, and we have a lot also, like in Chile, in Brazil, we do have a lot of very interesting strains in different sources and so on. No?
Yes, I think that's our main natural richness, our strains.
Indeed, indeed, indeed. So there are some comments and questions coming out. Evelise said that at least for some genera, I lost here. Okay, from some genera, Bacillus, Penibacillus, Bucodera, Enterococcus, it's very difficult to differentiate soil from marine isolates regarding BGCs.
It's true. It's true. Yes, it's the same here. We are trying to identify some marine adaptation genes that maybe we can use just to have like evidence that these biosynthetic gene clusters are observed only in the marine environment. But it's very hard because, for example, for us, the number of marine genomes are very, very low comparing to the one that comes from soil or terrestrial sources. So that's why I encourage other authors to sequence more the marine environment, so we can compare our biosynthetic gene cluster, and hopefully in some time we can find and see a clear signature of what is marine and what is not marine. But for example, some chloroperoxidases and bromoxygenases, they are just found in the marine environment, and they are good candidates to see, okay, so this is a root of a marine strain.
This is more or less the comment that Evelise did, that hopefully sequence more genes, you give a better picture from that. No?
Yes, yes, that's our aim in the future, for sure.
Yes. Janira said also that some class of bacteriocins are easy to chemical synthesis. They do not require enzymes for the maturation in small structures.
Yes, that's true. That's the main difference. NRPSs are very long pathways. You need first the genes for the biosynthesis, and then there is other genes that are involved also in the modification. So the biosynthesis is really complex, and also the regulation of the biosynthesis is indeed more complex. So sometimes you can find a lot of biosynthetic gene clusters, but actually they are silent in laboratory conditions, and you don't see the expression. So there are a lot of work just to awaken this biosynthetic gene cluster, and that is to work with the regulation and also with small molecules that can also activate these biosynthetic gene clusters. So yes, the biosynthesis is really complex, but we believe that that is also translate into complex molecules that has a chemistry that has more value for the industry. So that's why we are focused on NRPSs.
Yeah, and this should be the speech for the pharma companies. This is the speech.
Yeah, wrote it down.
We have a question from Bea. Great talk, Agus. Can you tell us why it's so important to have high-quality genomes for comparative genomics in our research field?
Yes, so we always make the difficult questions, but this is a good one, because it was from our experience. I can say that we truly put an effort to pursue high quality genomes. There is just a vast burst in genome sequencing that now everyone can do it, right? And so you can find a lot of sequence in NCBI, and some of them are really, really, really in drafts, but in very bad drafts. So when you download it and you try to compare in terms of biosynthetic gene cluster, that they are these really long biosynthetic genes that are composed with different genes. So you have a lot of steps in the biosynthesis. You truly need a good quality genome and also a good assembly, because otherwise the biosynthetic gene cluster is being split and you can see just a piece of the puzzle and you actually don't know where the piece goes in the big puzzle, and it's truly difficult to interpretate the biosynthesis and how this cluster is involved in producing one molecule or another one. So I think that a message to everyone that is doing genomics is to actually try to pursue, even we know that you have to put more money into it because you need to pay for PacBio or Nanopore, but the combination of short reads assemblies like Illumina with the long reads assemblies like PacBio or Nanopore are truly needed to find like a good quality closed genome, and then with that good quality closed genome to perform another comparative genomics assays.
Mm-hmm. Good. You can see in the chat there is one question more from Janira. She's asking why have you read it? Why have you used any more than 98?
Yes.
To avoid the clones or every similar lineage is a question, or because they use the more than higher than 95 to confirm especially normally.
Yes, that's true. We used 98 because we have the aim to just replicate the strains. So if we have two strains that cluster together in the tree, we applied ANI just to see if they were redundant strains. Sometimes we have a strain that we download and the same researchers, after they sequence again and they have like another entry with a different name, and we have mostly the same genome, but with two different entries. So this was like a way for us to check if the genomes were actually the same. We were looking for identical genomes, and we were trying to replicate those synonyms genome. So without the taxonomic specifically if they can be the same species or not, or another strain of the same species, we were just doing ANI to corroborate that they were not redundant genomes. And if so, we just selected one, and the other one we discarded.
Mm-hmm.
We selected the best assembly.
Mm-hmm. Good. Okay.
But all those steps are better explained in the article. I have a little time to explain every part of the pipeline, but actually it's a lot more bigger than what I put here in the presentation. Mm-hmm. So if you're more interested, you can go to the paper, and if you have any more doubts, you can write to me or anyone in our team, and we can explain it to you a little bit more.
Mm-hmm. Good, very good. I think we don't have any other questions, only comments congratulating you for the talk.
Thank you.
Indeed, it was really nice and very clear. Also, the slides and so. I would like to thank you again for your availability to be here with us. And
thank you for the invitation, Alex.
No, it was great pleasure for us. I was sure that it would be nice, and indeed it is nice your work. So that thank you everyone to be with us here on this Friday. Have a nice weekend. We meet each other next Friday again, 11 a.m. here in Brazil. Agustina, if you can stay just a few minutes more, we cut the
yes, no problem.
Okay, so
Obrigado,
thank you
everyone. Thank you for
Obrigado.
listening.
Thank you.